This Week in Parasitism is brought to you by the American Society for Microbiology at microbeworld.org/TWiP. This Week in Parasitism, episode number 47, recorded on November 27th, 2012. Hi, everyone. It's Vincent Racaniello, and joining me is Dickson Despommier.
Hello, Vincent.
Buongiorno, Dickson.
Buongiorno, Vincent.
How are you?
I'm well.
It's been a month since we last spoke, probably over a month. And we can use as an excuse Hurricane Sandy.
1.
And Italy.
2.
We blame Italy for everything, right?
Absolutely.
Which is where you were.
I was.
And I understand you unearthed some parasites in Italy?
No, we didn't look for parasites, actually.
I'm sorry.
They were in Paris.
Are you here every night?
Wednesdays.
Speaking of comedy.
Yes.
How's your comedic son doing?
He's doing great, actually. He's doing very, very well. For the listeners out there who don't know my son, I would promise that you do know my son, but you don't know him personally. You know him from the Aflac commercials, particularly the one involving the Aflac pigeon. He is the voice of the Aflac pigeon.
You must be such a proud father.
I am bursting at the seams.
Well, that's good. He actually recorded some intros and outros for us.
He did.
Which I should use one of these days.
You really should. You have to pay him a royalty, though, if you did.
Forget it. There's nothing to pay.
That's right.
Well, it's good to be back.
Yep.
Are you pleased, or you don't care one way or the other?
No, Vince, it's good to be back. It's very good to be back, in fact. When you leave your home country and you travel and you enjoy the cuisines of wherever you are, some time in the trip you start to miss the familiarity of where you came from. and this trip was just long enough. So 2.5 weeks, and it was a wonderful tour of places like Perugia and Florence.
And you did some
Firenze.
talking about vertical farms, right?
Yeah, well, we talked about that in Perugia, and then toured in Florence and Venice.
All right, now you're back for a few weeks.
At least.
And then you're off again. So let's get in a couple of TWiPs before you go.
You've got that right.
TWiP the light fantastic.
Exactly.
So today I thought we would discuss a paper.
Yes.
That I discovered before you left.
I can see it.
I had hoped to get it before you left, but for some reason something came up that Monday. I think I had to go down to the Barrier Island and take care of my swamped house.
Exactly. Right.
Something like that.
Yep.
Which
yes,
continues to be pain.
Too bad, Vince.
Well, the electrical is messed up.
Oh, dear. Salt water will do that.
Yeah, when it gets in the wires, they short out when you turn the power back on.
They do. They do.
And so
there's still one tunnel from Brooklyn to Manhattan that has not been opened because of that. Yeah,
which one? Right. Brooklyn Battery?
It's the R line.
Oh, for the train, you mean?
Yeah, the R line.
So today we can get to this paper. It's a
good
+1 paper.
Right.
And it's from a collaborative group, but the last author is Max Niebert.
Yes.
Who I know for many years.
Do you?
He's a virologist at Harvard. And
he's a virologist at
was trained by Bernie Fields.
Right.
And I've known for a long time. He's worked on reoviruses, which, of course, are double-stranded RNA viruses of mammals. But now he has turned his attention to the double-stranded RNA viruses that infect a number of parasitic protozoans.
Isn't that interesting?
Like Trichomonas, Leishmania, Giardia, Plasmodium, Entamoeba,
right.
Naegleria, Eimeria, Cryptosporidium, and Babesia.
Wow.
I've talked about most of those except Eimeria.
True. That's a chicken infection.
And Babesia, I think we didn't talk about.
Babesia is it's a zoonotic infection carried by ticks. It was I think, if I'm not mistaken, the very first microbe to be shown to be transmitted by a vector, because Theobald Smith.
Shipping fever?
No. Shipping fever is brucellosis. That's not
What's
transmitted.
I remember this story. You told this.
Yeah, I did. This is the organism transmitted by the Lone Star tick.
Okay.
And even though you're right that cattle get infected with brucellosis, they also get infected with babesia. And people without spleens get infected with babesia and die. There's no cure for it. So there's huge warning signs up on places like Nantucket and other island communities where there are lots of deer, there are lots of ticks. And it says, if you're missing your spleen, don't show up,
So
please.
it's in deer, and we get it from
Yes.
deer via ticks.
That's right. And in mice as well.
And Eimeria is a chicken parasite.
It is. It's the curse of Frank Perdueism.
What does it do to them? It
makes them
not
skinny.
gain weight, right?
That's exactly right. That's exactly right. So a lot of effort has been devoted to trying to come up with anti-Eimeria-like antibiotics.
But it wasn't worth an episode, right?
We could,
If there's a paper one day, who knows?
we could revisit this
We could.
subject. There's no harm in it whatsoever.
The name of this paper is Endobiont Viruses Sensed by the Human Host Beyond Conventional Antiparasitic Therapy. What is an endobiont? I don't know.
I've never used the term myself, but I presume that it means that it's a virus that infects cells that infect humans, just by judging for the title.
These are all ectoparasites, aren't they?
And yeah, endoparasites, Vince.
Well,
Endo.
do they get into the cells?
They do. Every one of the ones except...
Trichomonas?
No, let's go down the list again.
Trichomonas?
Let's see this list. Trichomonas is not an intracellular parasite.
All right. Leishmania?
Leishmania is definitely an intracellular parasite.
Giardia?
Not an intracellular parasite.
Plasmodium is.
Definitely.
Even I know that.
This is right, but it's not listed here, is it?
Yes.
It is. There is a... I didn't realize that they had an endobiont virus.
Plasmodium?
I had no
Yeah.
idea that Plasmodium harbored such an item. There's a lot of older work by C.C. Wang out at the University of California in San Francisco who
He did the
championed-
Leishmania virus.
He did. He did, and he did some wonderful work on it. So that's how come I come to know about this, but I didn't realize it had such a wide range of host species to infect.
Entamoeba?
That's not an intracellular
Okay.
parasite.
Eimeria?
Yes, it is an intracellular parasite.
Neglaria?
Neglaria is not an intracellular parasite.
Crypto?
Crypto is an intracellular parasite.
Babesia.
Definitely an
It's
intracellular
a mix.
parasite. So
So anyway, all those
It's a
are
mixture.
protozoa, right?
all those are protozoans.
And they have these double-stranded RNA viruses.
Correct.
And so Max has turned his attention to them.
How about that?
And in fact, when I visited, I think it was last fall, he told me this story,
Nice.
and now it's published.
Excellent.
Interestingly, these double-stranded RNA viruses, Dickson,
Yes.
do not seem to harm their protozoan hosts.
This is a mystery. This is absolutely right. That's what I remember from C.C. Wang's work, in that they're not doing anything obvious.
Right.
So what are they doing if they're not doing anything obvious? They're not killing the parasite. Too bad.
And they don't infect human cells either.
No. It's a shame they're not killing the parasites, because they could be used as weapons against the parasites, then, couldn't they? But they're not.
Do you think they're of benefit to the parasite?
Well, perhaps this paper will address that issue.
Didn't we do a paper once where the presence of the Leishmania virus made it more virulent? Is that, am I remembering? Maybe we did it on TWiP.
No, I
And we
think
had you as a guest.
as an itinerant. Yeah, no, that's right. That is exactly right. And we've covered other endosymbionts, Wolbachia, for instance, as a bacteria, which has a dramatic effect on pathogenicity.
Oh, yeah.
So
We just talked about Wolbachia on
yes.
TWiP.
Aha.
You know, in Drosophila,
Yes.
Wolbachia is good for the female.
Is it?
It makes them lay more eggs.
Get out of here.
It's really, they figured this out.
Really?
Yeah.
Why would that work like that?
It increases
to make more Wolbachia.
mitosis of germ cells, and it decreases apoptosis. So they make more eggs.
Wow.
So it's a maternally beneficial endosymbiont.
Well, it means that it can spread itself among more offspring, then, can't it?
Yeah.
So that works in favor of Wolbachia.
Now, in this paper, they focus on Trichomonas vaginalis,
Ah, yes.
perhaps the most ubiquitously spread protozoan parasite.
It's an anaerobe, Vince.
Yeah,
It was made.
lives in the vaginal tract, right?
That's true.
But there's not a lot of air, correct?
No, and it has a very unusual
Actually, I shouldn't say that. There probably is some air.
electron transfer system that involves a unique structure called the hydrogenosome.
Yeah, that's right. Which René Dubos discovered, right?
No, Miklos Miller, actually.
Miklos Miller.
Right.
It was close.
Yeah, they were both at Rockefeller.
Yeah, sure. Close. I said close.
They were close in proximity, but
Yeah.
not in interests.
It says here, in 2005, there were 248 million men and women infected worldwide.
This is true.
Do you remember when we did an episode on Trichomonas,
Yes,
by
we
the
did.
way?
Yes, of course.
And you mentioned besides the sexual mode of transmission,
Yes.
there are non-sexual ways as well. And
There
one of
are.
the docs wrote us and said he didn't know that. And he had to
Interesting.
apologize to a patient who he had said, you got it through sex? And they said no.
If you're a female baby and you're born of a mother who has trichomonas infection, it's very possible to catch it from the mother on the way out of the birth canal.
Is that called vertical transmission?
What would that be called?
It's vertical, mother to baby.
Yeah, sure. Not in utero, but just
Yeah, as the birth occurs, yeah.
after utero.
And then the baby may harbor that for a
A
while.
long time asymptomatically.
Yeah.
And men, mostly asymptomatic,
Mm-hmm.
which is why it's so rapidly transmitted, because nobody knows they have it.
Right.
So you can't blame them for transmitting it, basically.
So they say that by PCR,
Mm-hmm.
over 50% trichomonas positive in some urban centers of the U.S. So very frequent, even without symptoms, as you say.
Right.
I've lost my voice from lecturing for an hour and a half. We're
Let's
both
go-
a little...
Let's go find it.
Right. Is there a finder's fee?
Well, maybe.
Now, the interesting thing here, trichomonas infection frequently recurs and coincides with a disturbed vaginal microflora syndrome known as bacterial vaginosis,
Right.
and increased susceptibility to other infections like HIV,
That's
human papillomavirus,
Or yeast.
cancers. So there are lots of side effects.
That's
And
right.
there are also reproductive complications,
This is also true.
preterm delivery, pelvic inflammatory disease, infertility, pregnancy loss. All, and look, mother to child transmission, which is
There
what
you go.
we just talked about.
That's right. We did say that.
$26 billion in costs in the U.S. alone with
Wow.
preterm issues.
Wow.
And so they're trying to understand why this happens.
Sure.
Now, you can treat it with,
Metronidazole,
right,
which treats all anaerobes, by the way, including bacterial anaerobes. Very interestingly.
and what they say here is that a review of all the clinical trials of treatment of trich with metronidazole says,
Yes.
even though it seems to clear the infection,
Yep.
it doesn't get rid of the side effects, especially
Ah.
the pregnancy issues.
That's right.
So there's something else going on that they wanted to get at here.
Right.
You following me so far, Dickson?
So far, I'm following you.
So metronidazole does good clearance, but doesn't get rid of the side effects.
Or the pathological effects.
That's right, the pathological
That's right.
side effects.
That's right. That's right.
So what they asked in this paper is, do the viruses have something to do with this?
Oh, my. Well, wait a minute, Vince. If the virus lives inside the trichomonas
Right.
and the drug clears the trichomonas, why would there be any virus left?
You want to wait and find out?
I do.
Okay.
I'm sure everyone out there is wondering the same question.
Just let me give you a clue.
I will take it.
When you treat with the drug, the trichs rupture.
They do.
Well, they may.
They could. But you
Yes.
said that the virus doesn't infect anything
It
else
doesn't
except
affect anything else.
trichomonas.
It does not, yeah.
So it still raises the question of whether or
Right.
not it's involved in the pathology, right?
Right.
Right.
Now, their idea here is that maybe the viruses are being sensed by the innate immune system of the host.
Ah.
And you sense double-stranded RNA, you make interferon and other cytokines. And
Which
that leads-
also elicit inflammation.
yeah, exactly. So maybe that's what's going on here.
Vince, that's... But wouldn't it... Wait a minute. I've always been led to believe that it's very difficult to handle RNA because there's a ton of RNases out there, right? Even on your fingers. That's why you have to heat up the glassware to the point of it destroys the
Yeah.
RNase so that you don't denature the RNAs as you're processing your northerns. So why would a double-stranded molecule of RNA survive in a zone which probably has tons of RNases floating around?
Mm-hmm.
That's a question.
Well, double-stranded is pretty resistant to all those nucleases.
And it's only double-stranded that elicits toll-like receptor responses for innate
No,
immunity?
there are certainly sensors of single-stranded RNA, but
Okay.
typically those are encountered in the cell.
Okay.
There is a toll-like receptor on the cell surface that could detect double-stranded RNA, or it could be detected intracellularly. But we jumped the gun, Dickson.
We did. Well,
It's okay.
it's my innate curiosity.
Innate. Ha ha.
Sorry.
You're a jokester.
I'm trying not to be here.
So the experimental design is pretty simple.
Right.
They have these cell cultures, human epithelial cell cultures, representing the three mucosal compartments of the reproductive tract: the endocervix, the ectocervix, and the vagina. Okay.
Right.
And they add to those Trichomonas, which are uninfected or infected with this virus,
Double-stranded.
Trichomonas virus.
Right.
Actually, do you want to know a little bit about this Trichomonas virus?
Absolutely, Vince.
Well, I can tell you very little about it.
Okay.
It is a member of the family Totiviridae. And the
Totiviridae?
Totiviridae, and it's a new genus called Trichomonas virus.
Huh.
They're non-cytopathic. They're have icosahedral capsids, and they have double-stranded RNA genomes.
Would you call them endosymbionts of the Trichomonas then?
I don't know.
Could you
be.
ever call a virus an endosymbiont?
Yeah, if it does good, right?
Well, we don't know what's going on here, do
I
we?
think you could call them an endosymbiont in some cases. I don't know about this because there's no evidence, but maybe they'll have evidence one day.
Right.
So if you add Trichomonas to these cell cultures,
Yes.
with or without virus, so you can get them without infection. So not every trick out there is infected,
Right.
but a lot of them are. And in fact, actually, that's a good point.
Can you cure it?
Hang on, sir. I'm sure you could. At least half of the primary clinical isolates of Trichomonas have these viruses in them.
Right. You've highlighted that, as a matter of
Yeah.
fact. In the
So I forgot to mention that. So
Uh-huh.
it's kind of a correlation, right?
You'd think so.
That, if you have a high, maybe that's in part responsible for this
So
side
they could
effect.
do this at a clinical level too, couldn't they? In the clinic.
How so, Dickson?
Well, they could take a swab,
Yeah.
detect the virus or not detect the virus, and then correlate that with the pathology of the patient.
Yeah, and also look for cytokines
Right.
representative of inflammation. Absolutely. So this is a cell culture experiment
Right,
here,
right.
and the results are positive, as you will see.
Of course.
And I think the next thing is to do exactly that sort of experiment,
Okay. All right.
which is difficult. It's a clinical experiment.
Well, not if you do translational research in the laboratory.
If you're used to it. Me, I wouldn't get involved in patients.
Right.
It's too much.
No, you'd need
Bother.
a lab here and a good OBGYN person there.
A
to
collaboration
correlate.
would be great. You get a protocol. Okay, give me samples, and they send you tubes with samples. That's great
Right.
because they deal with signing the people up.
Exactly. And they can evaluate the inflammation responses as well.
Yeah. Then you measure the cytokines. You look for the virus. Well,
Let's
that's
do
the way
it.
science should be. It should be collaborative.
That's right.
No lab can do everything anymore.
No, and in fact, just as an aside, I saw a program last night on Scientific American, sort of a Nova Today or something like that, which showed a scientist who enlists about 30,000 people to help him fold proteins.
Oh yeah, fold at home. Yeah,
And
distributed computing.
that's his science group that he uses.
This guy's at Stanford, right?
Exactly right. It was amazing to see who got involved in
My
this.
friend Mark Pelletier had him on his podcast
Oh,
once. Yeah.
it's fantastic.
It's very cool.
It is. And
Are
that's
you going
without
to
a
do it?
grant.
Are you going to join?
Don't know anything about folding proteins.
You know how to misfold them, though.
Yeah, I do. That I can do.
So when you add the Trichomonas to the cells, then you can measure cytokine production.
Sure.
That's pretty easy. You do ELISAs.
Absolutely.
Those you
Exactly.
buy a kit and you can buy it. So
Right.
they measure a number of
You
cytokines.
look for cytopathic effects.
No, they just measure the cytokines
Oh, I'm sorry.
by
Okay.
ELISA,
I see.
like interferon, various interleukins.
Okay.
Okay.
Yep.
And what they find is that in fact, pro-inflammatory cytokines are produced. All right.
Wow.
By
Only-
adding only when you add the trick with double-stranded RNA virus in it,
That's remarkable.
not without. There's no induction of cytokines. Okay. And this happens in all three of these cell types. Now, the positive control is to add double-stranded RNA to these cultures, and that, of course, induces lots of cytokine production. So the implication here is that, in fact, the double-stranded RNA virus is inducing the cytokines in these cultures.
Yep.
Isn't that cool?
It's very cool, Vince. Very
Now,
cool.
one of the sensors of double-stranded RNA is TLR3.
Okay.
All right?
Yep.
That is present both on the plasma membrane and in the endosome, the
Right.
inside of the endosome.
Right.
So if viruses are endocytosed, they will get sensed by TLR3.
Got it.
So they knock down TLR3 in these cells with small interfering RNAs. And now the effect of cytokine induction by adding the trichs with the virus is gone. It's rapidly, greatly reduced.
Wow.
So it suggests that
Wow.
the innate sensor, or at least
Wow.
TLR3, is doing this. Isn't that cool?
Pardon me. Yes, it is.
You have a cold, Dickson?
I think I have a little bit of a cold.
Is it a Trichomonas cold?
You can't have a cold from Trichomonas.
Thank you.
Sorry.
All right, so cytokines are produced and they're TLR3 dependent.
Correct.
Now, your question about the antibiotic treatment.
Can I cure the RNA
Yeah, why
infection?
why is it that if you treat the tricks with antibiotics, would there still be some RNA virus around?
Right. Or wouldn't they be, or would they be degraded?
Right. So they do an experiment where again, the same experiment we've talked about. You take cell cultures and you add tricks with virus or without virus. And they have a third condition where they add antibiotic, the metronidazole.
Right.
In the cells treated with the antibiotic, the induction of cytokines is even greater. Because so they think they're breaking open the tricks,
I see.
and the RNA viruses are coming out.
Got it.
What's an experiment you would do? Now, here you think these RNA viruses are stimulating, right, innate responses. What would you do that I haven't talked about so far? Anything, or you're fine with this?
No. You did the first level of experiments, which showed that at least it's possible to elicit innate immune responses from freed double-stranded
Well,
RNA.
infected versus uninfected. It's amplified by drug treatment.
And you said the positive control, was using just
Poly-IC.
poly-IC, and it does the same thing at the same level. So what else would
What else would you like to see?
What else would you like to see? Right. Well, you have to look further down the chain of command for the innate immune response. Namely, is interferon actually produced during this?
They looked at that.
And
Interferon is produced. Yeah.
It is produced. So the whole chain of command.
Yeah. They actually look at
Is
some of the intermediaries, and it's all activated. It all makes perfect sense. It's an interferon response. Yeah. So here, let me repeat to you what was done. Tricks with and without virus,
Right.
with and without metronidazole.
Right.
Poly-IC is a control.
Right.
Anything you'd like to see there? You can say no.
I think those experiments stand pretty well by themselves.
They purify the virus particles
Oh, okay.
and put those on the
Oh,
cells.
okay.
And they just add the virus particles to these human epithelial cell cultures.
These are encapsulated virus
Yeah.
particles.
I remember they don't infect the cells,
No, they don't,
but yet the cells make interferon when you add them to them.
because they're engulfed
Yeah, probably.
by panocytosis.
Yeah. In fact, they use an inhibitor of uptake. Berfeldin A. No, it's something else. Where is it? Signaling via cell-free virions. What's the inhibitor of uptake that they used? You remember what the drug is?
I don't think I ever knew what that drug was.
Well, then you can't forget it, can you?
It's hard to forget something you don't know.
So they add purified virions, and you get...
Okay.
And so they say that's in line with the idea that metronidazole is rupturing
Got
the trickies, the
Got it.
tricks, and letting virus particles out. They use an inhibitor of endocytosis, which is evading me at the time. And they show that is consistent with the virions being taken up by the cell by endocytosis. So you can imagine that in the vaginal mucosa, you have trichomonas that are
Right.
infected with viruses,
Sure. No, they're infected with.
double-stranded RNA viruses.
Wait a minute.
The
say that.
trichomonas.
The trichomonas are infected.
And they release periodically these viruses, and then they get endocytosed by the mucosal cells, and then they make inflammatory cytokines, and that makes side effects. Perfect. And then if you treat with metronidazole,
Yes.
makes it worse. it
Right.
ruptures the parasites, but the virus particles are still there to signal.
So is there a way of inhibiting the innate immune response locally?
Yeah, you could look for inhibitors and use that in conjunction with metronidazole, right?
But there's another way to do all of this, and that is to change the pH of the vaginal tract.
You think?
Yeah,
And what would that do to the trich?
it would wash them out. They exist in basic pH, and if you use a vinegar douche, for instance, this is recommended for women who are pregnant who can't take metronidazole because they're afraid of teratogenic effects. They still have to be treated because this is an awful infection to have to endure during that time. And so they use vinegar douche as a result. And it lowers the pH and it makes it unacceptable to the parasite and it disappears.
You think that they might still rupture and release viruses?
They of course they would. So then
That would
you'd
be a
have
problem,
to.
yeah.
Well, you have to do something else in addition, right?
So I think your idea of dampening the inflammatory response would be a good idea. Of course, first, as you said earlier, we have to do these studies in people and make sure.
Yeah. So here's
Right.
another question I have then: if inflammation is one of the common causes of trichomoniasis with the double-stranded RNA, and the inflammation is intense enough, does this breach the cellular barriers to allow HIV to enter?
Yeah. Well, they say here, the last sentence of the paper, it is conceivable that virus released by dying protozoa during conventional antibiotic treatment fuels pathogenic inflammatory responses linked to preterm birth and facilitating risk of HIV and other sexually transmitted infections.
So there you have it. So this is a very important paper then, Vince.
Yeah. So that's why the title is, a new paradigm. What is it?
Right.
Beyond conventional antiparasitic therapy. So you don't just need metronidazole, but you're going to have to add,
Right.
an inhibitor of inflammation.
Sure. I mean, there are other infections like that where when you kill them, you get a tremendous inflammation as a result, right? Like cysticercosis, for instance. And so you have to temper that response as well.
Remind us about cysticercosis.
This is the juvenile form of the tapeworm that gets into your brain.
Okay.
And then eventually when it dies, it releases its hold on your immune system, and then you get collateral damage. So to avoid collateral damage during treatment, they give steroids. I don't know what would happen if you gave steroids in this case.
It might be suppressive,
Might.
right? But, in the case of dengue, no, West Nile, I mean,
Okay.
it's supposed to use
That's
steroids, so I don't know.
No, they said
But there are
don't
other
use it.
ways. There are other inhibitors of
Yeah,
sensing that you could look into. I mean, this is a ways down the road, but
it is. Well, this just points up the fact that we have to revise our textbook because
Yeah.
this information needs to be out there,
Yeah,
and it's
sure.
not yet. And this is a good example of the kind of information that we.
well, the cool thing here, I think, is that here we have a protozoal virus, right,
Yeah.
which doesn't infect the mammalian host, yet the mammalian host has this exquisite sensing mechanism for double-stranded RNA, so you get collateral damage.
Right.
Right?
So here's another suggestion, Vince. What if, along with metronidazole, you gave an intravaginal suppository which released RNase?
It would have to, well, that would work if the RNA was naked.
How do you cut up double-stranded RNA?
Well, there are double-stranded nucleases, but what if it's protected by the viral capsid, though, and it gets into the cell? That wouldn't work.
No, it wouldn't work.
I think the anti-inflammatory is probably the best.
Yeah, the dexamethasone
Approach,
or
right.
something like that. Yeah.
And remember, once you get rid of most of the trips,
Tricks.
you're not tricks, tricks and trips.
That's okay. They're both
You-
flagellated protozoa.
You don't have more virus production.
No, you don't.
So if you can use the antibiotic plus immunosuppressant.
The best way to do this is to do a vinegar douche wash first,
Yeah.
which eliminates the protozoan from the vaginal tract, and there are probably some residual protozoans left, followed by metronidazole and the anti-inflammatory. Bingo.
Would you like to do the clinical trial? No, you're retired,
Not
right?
clinical. I was
You're
never
not a clinician.
I was never a clinician anyway.
Well,
so.
people will be doing this.
Yeah.
And in fact, I should
Yeah.
mention the authors on this paper.
Please do.
Some of them actually are clinicians.
Well, you got to get your Trichomonas from someplace.
I've lost my...
In fact, I want you to know, Vince, that the first recorded case of metronidazole resistant trichomoniasis was isolated from a patient at Columbia.
Oh, it's resistant, eh?
Yeah, there was the guy's name was Miller. I'm trying to remember his first name. Dr. Miller, who was a friend of mine. It was a long time ago, so I can't recall his
Was he
first
in
name.
genetics department?
No, he was in OBGYN. He was the admitting attending. And they had a woman with acute trichomoniasis. They treated her with metronidazole 3 different times. The protozoan would not respond to the drug. I then walked the culture over to Rockefeller University and handed it to Miklos Mueller. And Miklos was a good friend of mine because I did my postdoc work with him over there. And he cultured it out and confirmed the fact that it was resistant to metronidazole. Became the first documented
And
case.
what's the mechanism of resistance?
Good question.
Never figured it out. Huh?
Well, he might have, but I didn't follow that literature.
The authors are Fichorova, Lee, Yamamoto, Takagi, Hayes, Goodman, Chipalotrea, Buck, Murray, Kula, Beach, Singh, and Niebuhr. It's a collaboration between Harvard Medical School, SUNY, Syracuse, a couple of medical centers, and basic science departments.
Right.
I'm surprised that the tricks,
No, tricks.
Trichomonas are not sensed themselves by the innate immune system. I guess there's no point.
They're not intracellular parasites, Vince. So they live in the mucus. They live underneath the mucus of the
Epithelium.
Of the epithelium.
Yeah.
That's right.
I guess, okay. But there are plasma membrane sensors, not a lot of them. There's TLR3.
But they don't secrete anything that would
Yeah, that's
cause
true. It
them
wouldn't be
to.
sensed. But there's some TLRs that detect protein, glycoprotein,
Yeah.
lipopolysaccharide on the surface. So it's weird, right?
Maybe the organism produces an inhibitor of those responses. A lot of these organisms do that. So, maybe they modify their environment to suit themselves.
Want to do some email?
You bet.
I have a letter here from a senator.
Really? A senator?
Yeah, look. It's on the United States Senate.
It's a senator.
It's not to us. It's to my son.
Oh, really?
Yeah. So my son, for one of his merit badges, had to write a letter to a senator
Aha.
like a year ago. So he
So
said, Dad, what do I write about?
Right.
I said, ask him why the NIH budget. Tell him that the NIH budget has to go up.
Right. So,
So-
which senator was this, Vince?
So this was Lautenberg.
That's a New Jersey
New Jersey.
senator. Yeah.
So he wrote back, Dear Mr. Racaniello, thank you for contacting me about biomedical research. I appreciate hearing from you on this important issue. I strongly support increased funding for scientific research, and I have consistently voted to increase the NIH budget throughout my time in the Senate. Since the beginning of 2009, I have supported the nearly $103 billion allocated to NIH through the American Recovery Act and appropriation bills for fiscal years 2009 through 12. This funding is critical to ensure that our nation's investment in life-saving research keeps up with biomedical inflation. And he goes on to detail all the money he approved. Throughout my time in the Senate, I have been a consistent supporter of the NIH, the CDC, and other medical research and outreach programs. Please be assured that I will continue to support the NIH and will keep your views in mind should this issue come before the Senate. Thank you again for contacting me. I should write him a letter and say,
Thank you for the thank you.
what is your support of TWiP, and TWiM?
Right. No,
I'll
that
never
wouldn't be appropriate.
Never
I don't
get an
think,
answer. Yeah, because it's self-serving.
I think a little bit, yeah.
No, I'm teaching. I'm teaching, Dickson.
And so am I, Vince.
Well, yes, we're doing this together.
That's true.
I have a letter from Lisbeth, who is from Brazil, or from South America.
Okay.
And this is from September. Tomorrow starts the 18th International Congress for Tropical Medicine and Malaria here in Rio de Janeiro.
Aha.
I read that Peter Hotez will be participating in a roundtable session on what is the future role of academic journals in research control and prevention of tropical diseases. I myself will be presenting partial results of my graduate project on the zoonotic potential of Giardia duodenalis in the past. Dr. Racaniello recently participated in the Brazilian Virology Society meeting in São Paulo. But what about Dr. Despommier? It would have been awesome meeting you both here. But, well, too many congresses, too little time, right? And
That's
money
true.
for that matter.
Yeah, money is the big issue here.
All the best.
And time.
He's thinking of us, Dick.
That's very nice. That's very nice. I would love to do a live TWiP TWiP show.
From a parasitology meeting, right?
Down in Brazil, this would be fabulous.
Someday I
Especially
think so.
right about now, as I'm looking out the window, it's snowing like heck.
Is it snowing?
It's snowing, Vince. That's not a cloud that settled into New York. That's snow out there.
Sophie writes, hello, Twipsters.
TWiPsters.
I like that.
We are TWiPsters.
I have a pick of the week, Window Farms,
Oh, great.
a nonprofit vertical gardening
Yeah.
project.
I know it.
How to make your own vertical window garden.
Good stuff.
If you want a full description, listen to Britta Riley's TED Talk.
Aha.
I can't do it justice. I don't know if this is a fitting pick, as it's more a Dickson pick than a parasitology pick, but I think it is amazing. I'm definitely doing this.
You can't make me laugh now, Vince, because I have a cold.
Keep up the good work. You see how people adore you?
Stop it. They don't adore me. They adore the idea of farming indoors, though. They do
Anyway,
that.
They write and they talk about you.
They do. That's flattering.
Now we have a couple of emails from Peter about Toxo.
Ah.
Dear Vincent and Dickson, I would like to point out that the female owners of cats more prone to suicide article mentioned in TWiP 43 is rather misleading. The study mentioned made no mention of cat ownership, and men were not included in the study, as it was only mothers who were included in the cohort.
Mm-hmm.
Most human infections are from contaminated meat, not from handling infected cats. If a cat is infected with toxogandii, the oocysts are only shed for a short period of time, typically less than 14 days, before the cat's immune system stops oocyst production. The cat should then be immune to further infection. Dr. Postolash has noted limitations to the study, such as the inability to determine the cause of the suicidal behavior. Could the high rate of infection reported in France be due to the popularity there of rare meat?
It certainly is, and I think John Boothroyd, having been a guest on our show, confirms that entirely.
We asked him point blank.
We did.
He said, nah.
He didn't say that. He did say nah.
And
That's
so
right.
Peter sends some more links on cats and toxoplasmosis.
Right.
The next one is also from Peter, but this is about nematomorphs.
Nematomorph.
Thank you for a fascinating and informative TWiP. I thoroughly enjoyed the linking of parasitism and ecology. Remember?
I do. Of course I do.
Following your request for listeners to find more information on the life cycle of nematomorphs, I did some reading and found that many nematomorph species use a paratenic or transport host in which the larva form cysts and do not develop further until the paratenic host is eaten by a scavenger or predator. Crickets are generally omnivorous, but some species are carnivorous.
They are.
A wide range of species can be used as paratenic hosts, including flatworms, snails, and some nematomorph species make the transition from water to land by forming cysts in aquatic insect larvae,
Sure.
with the cysts surviving the host's metamorphosis to a flying adult, which can then convey them to land. Adult nematomorphs are short-lived and do not feed. They die after mating. One species of nematomorph has been discovered from Kenya that is parthenogenic and lacks males. Some sites I looked at mention the morphological similarity of the larval nematomorphs to some adult marine worms. And he sends some references.
Right.
Came across a bit more information on how nematomorph worms influence insect behavior. David Byron, Frederick Thomas, and colleagues at the Laboratory of Genetics and Evolution of Infectious Diseases at France's National Scientific Research Center in Montpellier, France, have found the worms produce proteins that mimic some found in the insect's nervous system. Somehow these prompt the insect to jump into water, allowing the adult worm to swim away.
Amazing.
And he provides a National Geographic and a YouTube link.
Wow.
Amazing.
It is.
Mimicry at the protein level.
Indeed.
The next one is from Tommy. Now, Tommy is a big fan.
Good.
And I will actually, at the end, we'll tell you who Tommy is. He writes, Dear Vincent and Dickson, I really enjoyed your episode on the horsehair worm and food web ecology. In case you haven't already found it, here's a review on the biology of nematomorphs, which you might find useful. Also, I was pleasantly surprised to hear you talk about the New Zealand cockle trematode system. Those papers you mentioned originated from the lab where I did my PhD.
Wow.
In fact, my PhD thesis and a small part of my postdoc work consisted of research which followed up on some of the questions which were raised by those initial studies you cited, delving deeper into the ecology of that host parasite system. And he sends a small selection of publications that pertain to the cockle trematode system.
Nice.
All sorts of cool stuff, and he's the first author on all of them.
That's
He's done a lot of work on this system. Vincent was wondering how the cockles become infected with the trematodes. I can answer that, as I conducted and came up with the protocols for experimental infections of those cockles and spent many, many hours observing and documenting the process. Are you enjoying this, Dickson?
I'm relishing.
The free-living Cercariae are initially sucked in through the cockle's inhalant siphon. Once inside the cockle's mantle cavity, they immediately cling to the foot and begin penetrating into the muscle. Once within the foot, they then migrate through the muscular tissue for some time, a few hours at most, before insisting at a suitable spot, usually the tip of the foot. During one of my observation periods, I recorded some footage of the Cercariae.
Footage? Did you say footage?
Yeah.
Was that like a pun on... Sorry, we just jump on things like that because our own lives are so boring, we have to find humor in other things.
Speak for yourself. I do podcasts. My lives are fascinating. My life is fascinating. I said my lives are fascinating. I recorded some footage of the Cercariae penetrating the foot tissue, the link to which I recall I've sent to you in another email, but to save you the trouble of wading through masses of old email. Wading, you get it?
You got it,
It's a YouTube video of this penetration.
Oh, I love it.
It's cool.
Great.
P.S. I really appreciate all the effort and time which must go into making all the This Weekend podcast series and the role they play in educating the public about the unseen and overlooked majority of life on this planet. I am the writer and co-administrator of the Parasite of the Day blog, where
great.
I write about newly published research on parasites that I happen to come across in a manner that is accessible to the general public. I consider that my small contribution as a scientist for public education and promotion about parasites, parasitism, and parasitology.
Wonderful.
The Daily Parasite.
We'll have to... That's my pick of the week. That just became my pick of the week.
It's actually a site that we have mentioned before because Susan
we have?
Perkins.
Okay.
Do you know Susan Perkins?
I know the name.
She's at the Museum of Natural History here.
Yeah, exactly.
Did you know that?
I did.
And she had done it for a while, and then Tommy jumped in. Yes, she's a curator, program officer in the systematic biology and biodiversity inventories at the NSF. I'm also a curator at the American Museum of Natural History.
Nice.
We should get her on the show, Dickson. She's right here in New York, and Tommy, too.
You bet.
These people have things to contribute. We've already contributed all we know.
Yes, we're totally tapped out here.
I don't think so, but we should. These two would be very interesting.
I would love that.
All right. The next one is from Robin. Mm-hmm. It is, he sends us what is called a conversation with Robert Sapolsky. Do you know who Robert Sapolsky is?
I probably should, but I don't.
He is professor of biological sciences at Stanford University and has written books including A Primate's Memoir and Zebras Don't Get Ulcers.
Ah, that's why I don't know him.
So a quote here. This is Robert Sapolsky talking.
Yes.
A few years ago, I sat down with a couple of the toxo docs over in our hospital who do the toxo testing in the OBGYN clinics. And they hadn't heard about this behavioral story. And
this was in Stanford. This is in Stanford, right?
I suppose.
Yeah,
Yes.
so guess who that might be over there?
Yeah, Boothroyd.
Or Jack Remington, basically.
Oh, yeah?
Yeah.
I hadn't heard about this behavioral study. I'm going on about how cool and unexpected it is.
Right.
Suddenly, one of them jumps up, flooded with 40-year-old memories, and says, I just remembered back when I was a resident, I was doing a surgical transplant rotation, and there was an older surgeon who said, if you ever get organs from a motorcycle accident death, check the organs for toxo. I don't know why, but you find a lot of toxo. And you could see this guy was having a rush of nostalgic memories. What's the bottom line on all this? Well, it depends. If you want to overcome some of your inhibitions, toxo might be a very good thing to have in your system. Not surprisingly, ever since we started studying toxo in my lab, every lab meeting, we sit around speculating about which people in the lab are toxo infected.
We used to call those type A personalities.
You want to know something utterly terrifying? Here's something terrifying and not surprising. Folks who know about toxo and its effect on behavior are in the U.S. military. They're interested in toxo. They're officially intrigued. And I would think that they would be intrigued studying a parasite that makes mammals perhaps do things that everything in their fiber normally tells them not to because it's dangerous. But suddenly with the parasite on board, the mammal is a little bit more likely to go and do it.
Oh, dear.
Anyway, I don't know if that's true at all. According to John Boothroyd, that's not true.
And he's at Stanford,
not for people
too.
Anyway.
He ought to know.
Don writes, on TWiP 42, you decreed the lack of a microbe world search engine. Would it be decreed or decried?
Decried, I think.
Well, here's a
You
little...
were lamenting.
We're lamenting. Well, here's a little used Google feature that can help. To find the TWiP episode about Toxoplasma, simply type this as a Google search. site:/microbeworld.org, Toxoplasma, quote, this week in parasitism, unquote. As long as the site allows the Google crawlers, the result is usually better than site search features. Looks like the TWiP you are looking for was number 12.
Sorry about that.
That's right. I have to spend hours editing now.
No, I tried to muffle that one.
I love TWiP, and TWiM. I've heard them all, some multiple times. But my
Wow.
favorite has to be TWiP, largely because of the happy accident of a world-class parasitologist and a world-class raconteur being embodied in your co-host. Dickson Despommier is without peer.
Woof. There
Dickson's,
go my hats.
Dickson's modest erudition and your nagging straight man routine make for a very entertaining show. Please don't think I am belittling the part you play. That you could do what you do so graciously, given all your accomplishments, is rare. Lesser men would let their egos get in the way. So I was talking about me now.
Indeed.
I don't have an ego.
It's all about you, Vince.
No. That I can also learn something is also much appreciated. Don't change a thing. Thank you, Don.
No, I that's very complimentary, by
This
the
is
way.
really what embodies TWiP.
I think, we make a great pair because I think you know the molecular biology much better than I do, and you're highly trained in that area. I was never highly trained in that area, by the way. So I'm not using that as an excuse, but I'm just saying that that's not my forte. My forte is once I know what the story is, I can relate that to other parts of things that I do know.
Yeah, you're very good at that.
So, that I don't know how I do that, and it's not an important issue of knowing why.
It's okay.
It just happens to come out like that, and so
Yeah.
I totally agree with his take on the way we interact because I've rarely been able to do this with anybody else. I must say, this is absolutely right.
You are without peer.
Well,
You are a world-class parasitologist and raconteur. I'm the nagging straight man. Although today was not quite the same.
No, not quite.
But that's all right.
That's a wonderful thing to say. Whether it's true or not,
beautiful, yeah.
I'll
have to put that on the website.
Absolutely.
Kyle writes, Dr. Despommier, I am a student doing post-bac work in pre-med and was intrigued by your discussion of a possible study of the contamination of dog-run soils by Toxocara canis.
Yes.
I'm designing a study in the Chicago land area to gather this data and was wondering if you could recommend a method of testing for eggs in the soil.
Yeah.
Is there any preferential method in your opinion? Also,
Yes.
I heard on your podcast that there is some graduate work being done in New York and was also wondering if they would want to compare data. Thank you for the great work.
Sure. Thank you.
Remember the guy in Brooklyn, Michael Walsh?
Yes.
We should connect them, right?
We should. The best method I can recommend is to do a zinc sulfate flotation method, which takes advantage of the fact that most things have a specific gravity in a particular concentration of zinc sulfate that sinks on centrifugation, but the eggs and cysts of parasites float.
Mm-hmm.
And so what you do is you fill the tube right to the top with the fluid after you've mixed all the sample together in this particular concentration of zinc sulfate and centrifuge it for 2 or 3 minutes. There's a well-established method online that you can access, I'm sure. And then at the end, you carefully take a cover slip of a slide and you touch the surface of the tube and you absorb all of the parasite stages that are now floating. And then you take the cover slip and put it on a regular microscope slide and look. And that's the best way to get all of the eggs and cysts of parasites to go to the top of the tube and everything else goes down to the bottom. It's an old technique, by the way. And it's one which I learned here in 1962 when I began my life as a parasitologist in the technology aspect of diagnosis. So I think zinc sulfate flotation is still a good one.
Next one is from Ronald. He writes, I love your TWiP and TWiM and am sharing a balance of them with students as time permits. My absolutely only complaint is that, as an old geezer, my hearing is not what it once was.
Oh,
For me, there are often times when a speaker's voice seems to almost disappear Sort of a mumble for me. I believe this is because you speak, and this is natural, as if you are speaking to someone sitting close to you. Well, Dickson's right across the desk from me, right?
Exactly, and vice versa.
I believe it would help if you each spoke as if you were speaking to an audience, maybe with a microphone and never allowing your mouth to point away from the audience, aiming the sound down instead of forward without losing the cordial atmosphere. Trivia, I know, and I doubt anyone else has described similar issues. Keep up the great work. It's great to have a chance to learn from your discussions. I know you spend a lot of time preparing these, and I am truly grateful. Enunciate is the word that has been escaping me, but I believe that is the core of my problem. Well, Ron, we do have mics right in front of our mouths.
We do.
Perhaps we do not enunciate enough. And if we tried, it would become less casual and conversational, right, Dickson?
I think that's true. It's like listening to the announcements in Grand Central Station for the trains. You can never understand them.
Nick writes, hi. Firstly, I'd like to say I'm a huge fan of your shows, TWiP, TWiV, and TWiM. I am a student in Perth, Australia.
Oh, right.
In the final year of my zoology major, first year of my microbiology major. I'm extremely interested in getting some experience in a lab to do with viruses, parasites, and microbiology. So I was wondering if there was any help or advice at all you could give me. Viruses, parasites, and microbiology.
In Australia?
The paper we talked about today.
Yeah.
Nick, here's my advice. Go up to a guy's lab or a gal's lab, knock on the door and say, you want to volunteer. You're happy to wash dishes. Just get in the door.
Yeah, that's right.
But if you could find a lab working on Trichomonas viruses or any of these double-stranded RNA viruses of protozoan parasites, that would fit the bill, right?
I would say.
But it doesn't really matter, Nick. Just basic microbiology, viruses.
Sure.
Get in a lab.
You bet.
And if they say, we don't have money, say, fine, I'll volunteer.
That's right. So, and I would also advise Nick to move from Perth, which you did your original work in, to another city. You've got plenty of choices in Australia. You've got Adelaide just down the road. Not such just down the road, but Perth is way out at the end of Australia. So if you go to Perth or to Melbourne, which has a great set of universities and institutes, particularly Walter and Eliza, which I've spent some time at, or move up the road to Sydney, or even in, and I'm really blocking on this now, the capital of Australia, Vince. Do you remember the capital of Australia?
Sydney?
No.
Melbourne?
No.
Canberra?
Canberra.
Canberra?
Canberra. Yeah, the national laboratories are part of the national laboratories are in Canberra. But you have other options too. You've got Brisbane, just up the coast, and lots of in the university.
How about Hendra?
You know, that's a virus, Vince. That I know. Come on
that's
now.
A suburb of a
It's a horse of a different virus. It's a virus of a different horse. Sorry.
The people in Australia are cringing that we don't know their capital.
No, I knew it. It didn't come off. It didn't roll off the tip of my tongue.
What's
But
the
when
capital
you said,
of the U.S.?
Come on.
It's the capital of Chile.
Santiago.
Good. Next one is from Amanda. She writes, good morning, Doctors Racaniello and Despommier. My name is Amanda, and I just love your TWiP podcast.
Thank you.
This article caught my eye, and I thought it would be of interest to both of you, considering that it seems to be about a virus parasitizing another virus. And this is a paper, an article by Ed Young in The Scientist, A Parasite's Parasites. It's about the new giant
The Mimi
virus discovered, the brand new one that has a Sputnik or an associated helper virus or parasite virus. And
they should call that the UU virus.
The virophage. Sorry, that's the word I'm looking for.
Virophage.
It's actually integrated into the genome of the big virus. We talked about this on TWiV.
We did.
And probably you even remember, Dickson, right?
Not only do I remember the name of the show.
What was it called?
Turducken.
Yeah, viral turducken. That was TWiV 206.
Viral turducken.
So, Amanda, if you like that sort of thing, check out TWiV. Also, a recent study on DNA methylation in Trichomonas. Wow. Did you know that, Dickson?
I don't know everything, Vince, and therefore I don't know
another
that.
Article
I certainly
in The
don't know
Scientist.
that one.
So The Scientist is a very good magazine of casual science articles. This is by Beth Marie Mole, Genomic Methylation Collector. Bucking the trend among DNA methylators, whose epigenetic marks on the genome tend to be stable, the parasite Trichinella spiralis accumulates methyl tags over time, according to a study published last week. It is also the first nematode found to carry a methylated genome.
I'll be darned.
Methylation is typically used to control transcription.
Sure.
Right? You methylate or you demethylate to open or close the chromatin, right?
It puts an X on the bottom of the nucleosome. That's what it does.
X marks the spot. So this is a paper. Maybe we should do this sometime. It's called Differential DNA Methylation in Discrete Developmental Stages of the Parasitic Nematode Trichinella spiralis.
And who did those studies?
Gao et al.
Who?
GAO.
Ah, China.
Let's see. I got the link right here. I think it's from China. Yeah, it is from.
They have a lot of trichinosis in China.
All Chinese authors. And what is the affiliation? Provisional PDF.
Okay. The provinces of Hunan and Yunnan in China have very high levels of infection in humans from trichinosis. They claim that they're getting it from not pork more, but from lamb, which in my view is a very unusual way of catching it because sheep are usually herbivores. But maybe they're feeding them scraps from the slaughterhouses.
It's the Zoonosis Research Center for the State Key Lab of Virology and Genetic Engineering, the Chinese Academy of Medical Sciences, Changchun, China. I don't know Changchun. Do you?
I don't, but if we looked it up on a map, I bet you it's either in Hainan or Yunnan.
Well, their collaborators are from Shenzhen. All right, I'll look it up right now. We can do that.
We have the magic of the internet at our fingertips, at our not-so-active fingertips.
It's the capital and largest city of the Jilin province, located in the northeast of China. Jilin.
Northeast. Above Beijing?
Yeah, I think so. Let's see. I got to zoom out here.
Wow.
Here. So here's Beijing down here, and here it is way up here.
That's very interesting, because on my website for trichinella.org, I have, under the epidemiology of trichinosis, I have a distribution map for all of the cases of trichinella in these two provinces. Well, maybe they've identified those two provinces as sort of hotspots that need to be worked on in order to try to come to grips with this still raging epidemic, I would call it.
Keep up the good work. Can't wait to hear another podcast. You guys keep my brain active when doing routine lab tasks. Amanda is a research technician at Massachusetts General Hospital.
Wow.
You know where that is, Dickson?
I do. It's in Boston.
Yep. All right, here's an email from our buddy Jim in Virginia.
Okay.
FYI for Dick, just to show where the subject is appearing with comments. Do you know /dot? Do you know what that is?
Let me get my glasses here.
/dot? Do you know what /dot is?
Probably don't.
Okay. Singapore builds first vertical vegetable farm. I know you know that story, but
I do.
/dot
I don't know that.
is a site where stories get posted and then people comment on them, and then depending on how many comments, they can rise or fall.
I see. Has that risen?
There are lots of comments about it.
Oh, that's good.
And a lot of people are talking about LEDs.
Ah, yes.
For example, LEDs give high electrical efficiency, but they aren't cost efficient even with the savings.
Not yet. And by the way, the Singapore vertical farm doesn't use LEDs. It's totally transparent.
There's quite a discussion here, Dickson. You should really weigh in.
I will.
Michael writes, I'm a long-retired biochemist who spent his career in public health. The two things that I miss the most are access to journals and the roundtable discussions that I held in my office every morning. I accomplished this by providing free coffee and then Danish. Soon my office was bursting with those wishing to talk about what they were doing, where they were having problems, and what they were going to do about them. These were not meetings in the normal sense, but rather verbal free-for-alls where many problems were resolved, many friends made, and aptitudes which were dormant came alive.
That sounds great, by the way. That's a wonderful vision that he's just presented. That's fantastic.
While I often worked with the biochemistry of microbes, I never had occasion to do any work with parasites other than see an interesting slide or specimen that someone wanted to display to everyone in sight. When I started listening to TWiP, I first thought it was the filling of an enormous cavern of personal ignorance that drew me towards your show. But then I realized it was the Socratic method that formed the backbone of your lectures, with Professor Racaniello so often playing the foil. Yes. And the general banter, it was almost like being back at my favorite time of the day at work.
Cool.
As I now live in a rural area and can no longer have such conversations with my old colleagues, this has truly gladdened my heart and filled in a missing part of my life that goes far beyond the knowledge provided.
Oh, that's wonderful.
While I realize that this is not your primary intent, it is a delightfully serendipitous collateral benefit. I would like to urge you to ignore those curmudgeons and martinets who demand a formal structure and pedantic presentation. You are at your best when you allow your personalities to show through.
You can see how much we've actually avoided that.
This is particularly significant to me as I had been in an undergraduate honors program that had about 40 professors and a dozen students. We were completely self-directed and met either one-on-one or in small groups with the profs. We got to know them as people, and as a result learned more and did more than we ever would have under more normal circumstances. It was unfortunate that this outstanding experiment was far too expensive to be broadened to the rest of the students. Before I wear out your patience, I would like to note that I listen to many such podcasts, but that yours is the only one that does not request financial assistance, which I usually provide. Your program is most worthy of some mechanism of recognition, so I thought perhaps a donation in your names and TWiP to be a possible method of recognition of your outstanding work. Would you please advise me as to your favorite charity or research facility in parasitology that you would prefer the donation to be made? Truly gratefully yours, Michael. We're working on a donation method for our podcast, and someday it'll be up.
We're running out of Danish.
Yes. That's very nice, Michael. Thank you.
That was a great letter, by the way. Great letter.
All right.
Where is he from?
It doesn't say.
Oh.
A couple more. Robert writes, Sirs, in September 1903, the British satirical magazine Punch published an Insomniac's Ode to the Students of Tropical Disease. And here it is. Men of science, you that dare beard the microbe in his lair, tracking through the jungly thickness, Afric's germ of sleeping sickness. Hear, oh, hear my parting plea, send a microbe home to me. Sometimes it is better not to get what you ask.
I think you would sleep forever, unfortunately.
Todd writes, thank you so much for the Parasitology podcast. I'm encouraging my children and nephews who will probably pursue health-related careers to listen to it, first because it is such an interesting fly-through of the related science, and secondly because I find you two so interesting. However, my family eats a whole lot of sushi, and I haven't been able to find the sushi episode on iTunes. Did that end up getting made? Regards, Todd.
It did.
Let's see. Let me use Don's trick to search for the sushi episode. So Don said, type this into Google. So
search.
I'm going to type in site, microbeworld.org, SUSHI, This Week in Parasitism.
You found it.
I got lots of hits, but I don't know what they mean.
Oh. We had one episode where we were discussing the ascarids of cetaceans.
We went over this. We did the sushi
We
episode.
We did the sushi episode, but maybe it was in conjunction with the ascaris episode.
Well, I'm getting the letters here.
And the Toxicara episode.
Yeah.
Because it's actually an aberrant nematode infection.
Well, I'll look through and put a link to the sushi episode in the show notes.
Sure.
Let's see.
In short, the sushi restaurants of the world are now much more aware of this problem than they were 20 years ago. And for the most part, as far as I've been led to believe, they flash freeze the sushi meat in liquid nitrogen and then defrost it right away. It does not affect the texture, kills the worms. Everybody's happy.
P.S. I had been imagining myself to be a sort of forager and would eat the semi-fermented pears that fell from my tree after cutting away the portions containing ants and worms and washing them a bit. After watching some YouTube videos about roundworm infections, I got a little bit afraid, since I don't know but that the pears had fallen onto soil or leaves that had traces of dog, opossum, squirrel, rabbit, raccoon, or bird feces. However, a professor friend of mine told me that our immune system kills roundworms for us, and it's only children who are at risk.
Not true. You better go back and revisit that one. That is not true.
We
Nope.
did an episode on roundworms, right?
Yeah, what your professor probably meant to say was, if these people grew up in endemic areas and acquired these parasites as children, by the time they are adults, their immune systems are now used to kicking out the worms, and they are less infected than the children, but they're still susceptible to the infections. So I wouldn't...
We talked about raccoon feces transmitting things to kids, right?
It's the Balis ascaris procyon.
right.
That's the name of that one. And it's part of the zoonotic aberrant infection route that we talked about.
PPS, since I also occasionally dip into alternative health websites, I wonder if you have ever podcasted related to the clean hypothesis of allergies and the alleged cures of asthma and other allergies from exposure to roundworms
Sure.
in controlled situations and hookworms in uncontrolled situations.
Yeah, we had some talk
did.
about that. We did.
Todd, you really go back through the archive at microbeworld.org/TWiP and you'll find them all.
And now I can announce that in the spring there will be a book on the subject called People, Parasites, and Plowshares, which I've just recently submitted to the editors of the Columbia University Press, and I have a whole chapter in there about this particular issue. So
Neat.
you'll be able to read more about
So
that.
we don't have to podcast anymore. We'll just publish your book and that'll be
Oh,
it.
Vince, I wouldn't go that far. No, the book may bomb, and I may have to podcast just
No,
to
I
get
think
the
it'll be
information out.
fine. I think a lot of people are interested in autoimmune disorders lately, and those
Oh,
of
you
us
bet.
who live somewhat dirty and untidy lives wonder if somehow environmental stimuli are keeping us robustly healthy.
Yeah, that's a valid point.
Also, bacteria, Todd, over on TWiM, my other podcast, we've talked about the role of bacteria in, for example, curbing asthma in humans.
Yeah.
And there's clearly a positive side to being infected, for sure. And that'll do it for email, Dickson.
That's great, Vince.
TWiP can be found on iTunes and at microbeworld.org/TWiP. If you do like TWiP, a good thing you can do is go over to iTunes and just give us 5 stars, and that helps us to stay prominent on iTunes. Of course, if you don't like us, you don't have to give us 5 stars.
Right.
But if you don't like us, you shouldn't go to
You
iTunes.
can give us 4 stars.
If you have questions or comments, we love to read them. You can send them to twip@twiv.tv. Dickson Despommier can be found at trichoneella.org, verticalfarm.com, and medicalecology.com. Thank you, Dickson,
Welcome again, Vincent.
for the banter.
It's a pleasure. Always a pleasure. As Alan Dove would say on TWiV.
He does say that always.
It's always a pleasure.
Always a pleasure.
But
Always a good time.
It is always a good time, and it is always a pleasure.
We'll get another one in before you go off to your next
Well,
destination.
I hope we get several more in.
When are
I'd
you
like
leaving
to.
again?
21st of December.
You could be gone for the holidays, eh?
Yeah.
going?
we're
Japan?
We're going to go to New Zealand.
Cool. All right. I'm Vincent Racaniello. I'll be here all month. You can find me at virology.ws. You've been listening to This Week in Parasitism. Thanks for joining us. We'll be back soon. Another TWiP is
It's parasitic.
parasitic.